Welcome, folks. It's 12 p.m., so we are going to go ahead and get started. Thank you so much for joining us today. This is the fourth of 5 webinars that we're hosting this winter-spring season. Our next presentation will be on June 1st at 12 p.m. and will feature plants along streams and coastlines, and will be the last webinar in this series, but we will join you all again in fall for another series of 3 webinars. So be on the lookout for that. I'll be dropping the link into the chat shortly for our events page, so you can sign up for that next webinar and know where to go to look for that fall series as well. If you aren't already in the know and would like to be subscribed to our monthly science and conservation newsletter, Go Botanical, you can sign up on our website and we will post announcements for the fall series there as well. So in a moment, I'll drop all those links into the chat for you, along with my email. This webinar is going to be recorded, and I'll be sending out that recording to you sometime in the next few days. Also, feel free to ask questions using the Q&A feature throughout this presentation. We'll be holding a general Q&A at the end as well. You can type questions into the chat box as throughout too. We'll keep an eye on both of those. We'll try to get to all your questions, but if we don't, you can follow up with me via email. So we're hosting today's session from our Botanical Research Center on Kauai, as well as our Fern Lab, which is located in our Conservation and Horticulture Center down in Lehigh Valley. And I'm going to hand it over to Dr. Nina Ronstedt, our Director of Science and Conservation, who will now tell you a bit about today's session.
Yeah, hello, thank you, Amanda, and aloha, and welcome to all of you. It's nice to see so many of you here. So this webinar is about ferns, and especially on oceanic islands. Ferns are really a major part of the forest and of the ecosystem, and often somewhat overlooked. We often talk about trees and beautiful and scented flowering plants, but ferns really have a very prominent role in maintaining the ecosystem as well. So we thought it would be really nice to focus a little bit in on that, and we are also lucky that we have some really good people to talk to us about it. Ken, who's worked on this and the flora of Kauai and Hawaii for several decades, and will give you a really nice introduction to what ferns are, and especially what ferns here in Hawaii are and on Kauai. And then we'll be following with Mike DeMotta, who will give you a tour of our Fern Lab, and we're one of the few botanical gardens that actually have prioritized to get a fern propagation up and running. And it's not the same as growing seeds. It's sometimes it's easy, sometimes it's a little more complicated, but we're very excited that we have a specialized fern lab. And unfortunately, we're gonna miss out on Ruth Agoroya today. She's a fern propagation expert that has been working with us for many years from Tallinn Botanical Garden, but she has unfortunately fallen ill. But Mike DeMotta will be able to answer many of the questions, and Ken, and if you have anything more specific, we can forward that to Ruth, and she can answer later on per email. So yeah, thanks for joining. Hope you will enjoy it. Back to you, Amanda.
All right, so we'll go ahead and get started. Ken Wood, our senior research biologist, is going to go ahead and share his screen and give you an overview of ferns.
Okay.
Are you there, Ken? Okay, great.
Can everyone hear me?
Yes, and we can see your PowerPoint as well.
Okay, super. Well, I'm very, very happy to give this presentation to those that are having an interest in ferns and lycophytes. Anyway, I'd like to just start out by saying that they are a very ancient race and they are worthy of attention. I've been a biologist here at the NTBG since 1982, so I guess that's 40 years. It's been quite a while. And I mostly worked with flowering plants, but I also focused on ferns and lycophytes here in Hawaii and in the South Pacific, and did lots of research in the Marquesas and Micronesia, Society Islands. But here we'll talk about the ferns and lycophytes of Hawaii. And when I call them an ancient race, here we have the lycophytes on this phylogenetic tree, somewhere around 420 million years ago. These are the beginning of vascular plants. Before that, we have the liverworts, mosses, hornworts. So the ferns evolved from the lycophytes, and that's somewhere around 360 million years ago. And from there, we get the gymnosperms, which we have no species, native species here in the Hawaiian Islands of gymnosperms, but angiosperms, of course, we have quite a few, being an isolated island and relatively old. Kauai is 5.1 million years old, and plants have been island hopping from previous islands that formed before Kauai. So collectively, we'll call the lycophytes and the ferns pteridophytes. Pteridophytes are broken down in Latin for feathered plants. Pteridus, pteris is feather, and phytes references plants. And they are very worthy of attention, although a lot of people do say that they all look alike, but certainly if you start to develop an eye for them, there are certain characters that you'll learn to differentiate different families and species. So the ferns are covering trees in wet forests, so that they're epiphytes. They're growing on the trees, and they'll cover terrestrial zones, and they'll dominate niches in dry and mesic forests. So they're all over the place. Certainly in wet forests, we find a great abundance. And as far as their breeding system goes, they're much different than seed plants. With the seed plants, as you know, the flowers will have their sexual exchange, and the seeds will form right there. And then once the seed is dropped, you'll get a mature plant from that seed. Whereas with ferns, there's two generations going on. There's spores that are called sporangia, either on the lower side of the fern leaf or on the edges, sometimes in hanging in strobiles on the lycophytes. And once the spores burst, they'll go ahead and spread over to develop into a gametophyte. And from there, you'll have both male and female parts in the gametophytes. And with a mature gametophyte and a little bit of water, they'll create a zygote, and from there you'll get this cycle from the zygote to a new sporophyte. So here, once again, the sporangia bursts open, the spores come out, the gametophyte is formed, there'll be male parts and female parts on the gametophyte. They'll do their exchange, and then you'll get the sporophyte. This image here, I wanted to demonstrate that the ferns did spread to the islands via wind, sometimes from continental areas where they'd get blown up into the stratosphere and work their way over to islands. But once on the islands, they would spread also by wind, but also we had a whole suite of these flightless birds or weak flying birds, many of them ducks, anatids, and other geese-like birds that roamed the forest before man took them out, because they were quite naive. But anyway, they would eat the ferns and drop them. They would spread by running around the spores through the forest that way, which is kind of interesting. Now, this slide here is going to start with demonstrating what some of the lycophytes are. As I mentioned, they're the oldest of the vascular plants. They have microphylls or single-veined microphylls on their branches. They produce strobili. Here you have the upright strobili on the Lycopodium venusulum. There's around 16 native taxa, and some of the genera in Hawaii include Hypersia, Lycopodium, Phlegmarioris, and Selaginella. Once again, here you see the phylogenetic tree where the Lycophytes begin 420 million years ago, and the ferns after that. A couple of examples of Lycophytes in Hawaii include Hypersia erosa, which is a terrestrial one, Hypersia arabescens, which we find often in bogs and along stream banks. Here we have some of the epiphytic ones, Phlegmarioris phyllanthus. Here you see the hanging strobili, which I talked about earlier, where the spores would be released. And this one here is an indigenous one, Phlegmarioris filiformis. It originally was known from all the islands, but was not known from Kauai until our own David Lawrence, Dr. Lawrence, discovered it in Wainiha Valley in the early 90s. And since then, we found just a few small colonies of them. So there are rare species out there that we try to map and keep track of. A couple of other examples of these primitive ferns include Selaginella arbuscula. This is a very common lycophyte. And here we have Celotum nutum, or moa, which means chicken because it resembles the tail of a chicken. Another primitive group is Ophioglossum, which here represented by the Polyphyllum. And this is kind of an extremophile in Hawaii. The Polyphyllum occurs in coastal zones that can be quite hot, and after rains, you can see them coming up here where water probably ponded. And so they'll come up and they'll have their ferns released from this sections of the strobili. Another very interesting primitive fern is the Botricium or Scriptidium. Botricium, we had one species called subbifoliatum, and it's been missing since 1940. We've been looking for it in areas where it was collected historically, but never have found it. It's kind of interesting, the sterile frond here on the left, and here's the sporophore right down here, and you can see the sporangia. I have a good example of the image of the Dowsifolium, which was missing from Samoa, and Tim Flynn, who's our curator of our PTBG herbarium, actually rediscovered it after not being seen in Samoa for a very long time. It's a difficult fern to see in forests, so you have to have a keen eye. But anyway, this is one that is missing, the subbifoliatum in Hawaii, and it's considered one of the holy grails or the missing ferns that we look for. And for those of you who like to explore Hawaii, keep your eyes open when you're in remote areas. If you see these sporophores emerging, that's a really good indicator that you have Botricium. Another primitive fern is the Miradia, and it can be a large fern. Here you see the base with the swollen stipes, and here's the stipules that form at the base, and you see the free veins forming, and once again you can see the Synagia here, where the spores will be released. And once they're mature, yeah, they'll split open and they'll shoot out. Another group of ferns in Hawaii is the Hymenophyllum. We have around 9 native taxa, and they're usually very delicate. They're considered filmy ferns. They're protective indusias, either two-lipped or they have a conical indusia. And some examples of genera here in Hawaii include Doryopterus, Crepitomanis, Hymenophilum, Vandenboschia. Some of them will have long creeping rhizomes. Some of them will have erect rhizomes. Here we have Crepitomanis parvulum. Many of you might know it as minutum, and the names do change, as we all know, as we learn more and more. Here's a good example of the cone-shaped indusia on the Calistopterus baldwinii. Here you see the erect rhizome as opposed to one that would be creeping and sending up its fronds. Hymenophilum lanceolatum, another interesting one that covers trees and is quite abundant in the Hawaiian Islands, including Hymenophyllum recurvum. These you'll see everywhere, and one that was known from the Big Island over to Oahu was Hymenophilum obtusum, and just recently we actually Shauna Walsh, one of our biologists, and myself rediscovered it, or discovered it for the first time here on Kauai. So we do make these island records on occasion. And not very long after Shauna had found it, we also had a kupu intern that's been working in Limahuli Preserve, which is our North Shore garden. Cassandra found another little colony of these. So just recently that they were found after several hundred years of botanizing. And there is a lot of area to look. Here you just see a good image of the summit of Kauai, looking out into some of the drainages, many, many cliffs, slopes covered in ferns and trees. And these are the bog areas on the summit. So you're not seeing forests in this picture, but we have lots of mesic forests and lots of cliff zones. Kauai, like I said, is 5.1 million years old, and it's got a highly variable physical geography, eroded over time. So we have canyons and dissected valleys, and we also have some of the finest bogs in the Hawaiian Islands. An interesting group are our matting ferns, and here you see Dichronopterus linearis. It's got its sorae with no indusia or protective covering. Some genera in Hawaii include Dichronopterus, Botricium, and Staghorn. I think it's interesting also, these matting ferns are refuge for visiting seabirds. So we have several shearwaters and petrels that come, usually in the springtime. They'll make their way back to the islands, and they'll find open areas where they can come in and land, and then they'll dig burrows underneath these matting ferns, and then they'll carry out their nesting procedure until around autumn time, when the young birds will fledge and fly out. So ferns can be a refuge for some of our seabirds. Here we have the Staghorn, which is endemic. Ziplotrigium pinnatum is an indigenous one. So when I say endemic, I'm meaning that it's restricted to either to the Hawaiian Islands, or if I say a single island endemic, I'm referencing that it only grows on one island and nowhere else in the world. Also of keen interest, which I demonstrate here, is the Sybodiaceae. We have Sybodium glaucum and several other species of Sybodium in Hawaii. Here you see the two-lipped indusia, where the spores will be released. We have other tree-like ferns called Satellaria. Now, Satellaria is an endemic genus. Satellaria only occurs in Hawaii. It's in the Blecnaceae family. Its sorae are linear along their costa, and there's 6 species endemic, and there's several taxa which are rheophytic. This is a rheophytic taxon, Satellaria unisora. This is one of the endemic ones that is endemic to Kauai, Satellaria wagneriana, named after the great 20th century teratologist Herb Wagner. Herb actually was a mentor of mine when I first started looking at ferns. Satellaria unisora is here with the spores, the sporangia being exposed along the costa here. Another group that is quite abundant, 27 species in Hawaii, is the Espleniaceae. And so we have an indusial flap that's formed on the lower side of the pinnae, which protects the sporangia before they're mature and spread their spores. There are several endangered ones, and I have them listed here. The Esplenium ethiopicum is an indigenous one, not endangered, but here's a group of other native Espleniums from the islands. You can see the names along the bottom there. An interesting one is the bird nest fern. Many of you probably know that, and it's cultivated. Esplenium schizophyllum is one of our more highly dissected ones, although we have, from the endemic subclad Diellia, we have some very highly divided Diellia or Espleniums. And here's an example. There's the Esplenium diolmanii here, and it was thought to be extinct, was last collected in the late 1800s. And a naturalist was the first person actually to rediscover this incredible fern. You see, it's very black, dark rachies, shiny rachies, and it's a very, very interesting fern. But anyway, there was 2 plants that were found by Laura Arnold back in 2002, and subsequently a team of NTBG biologists found a really nice colony of almost 100 in the middle of the canyon. And there's a quick story I'll try to throw in since I have a little bit more time and Ruth isn't going to be able to speak. The way that this Diellia was rediscovered was by just having an idea of what type of communities it was known from. It was collected by an early pioneer here named Val Knudsen, who found it in diverse to semi-diverse mesic forests. And I utilized Google Earth and other remote sensing imagery and covered Kauai and the canyon areas of Kauai and looked at the colorations of the forests and chose a spot. And oddly enough, to make a real long story short, when we did go to that site, we were able to, we actually found this very rare Esplenium occurring in that large number of almost 100 individuals. And since then, conservation efforts by the Talon Botanic Garden, which is Dr. Ruth, and NTBG, and our own plant extinction prevention program here, along with other DOFA members, have been working to protect certain areas where this plant occurs. They built a fence around it. Plants were cultivated, grown out in cultivation, and then reintroduced. And so we have them actually regenerating in their natural habitats. This is another one that was rediscovered about the same time. This one is Esplenium diolysiniatum. And so there was quite a few of them found in the middle of the canyon, as I demonstrated earlier, somewhere in this region. Here you can see some of the forest, but there's also a lot of bare areas. So utilizing those remote sensing imagery, I would go into these different patches, find areas that had a certain color. It's kind of like finding gold. You look for the color of a multitude of species and going to those sites and then looking in the understories, and that's how those plants were found. Another interesting group are the Pteridaceae. Here you have a false indusium. It's actually a part of the leaf blade folded over, and the sporangia are protected in this zone here until mature, and then the spores are spread. Doryopterus stachora, Pteris terminalis, which used to be Pteris excelsa, and then Doryopterus angelica, which was discovered in the 90s, described by Herb Wagner and myself. There's less than 100 individuals out there. This is another example of plants that we've been able to cultivate. Here we have Connie Now, who was working with NTBG at the time, was going out and collecting spores and growing them in our fern lab. And this shows how highly restricted. We would call this a narrow endemic, this Doryopterus angelica. So it's restricted to a very small area with less than 100 individuals, but right now we have quite a nice colony of them being reproduced and being reintroduced into protected areas. Polypodiaceae, round sorae, they're pretty small usually, but they can be somewhat larger. They'll have entire blades, as you see here, or they can be lobed. Here you see the anastomosing veins of this Microsorum, Hawaiians called peahi. So our Polypodiaceae. We have an endemic genus of Polypodiaceae called Adenophorus. And quite a few species. I put up a few of the interesting ones. Some are pendant, like the Hymenophyloides here. Some are sub-entire, like the Ha'alilioanus. We have a Tenella that's totally entire. I don't have it up. But we have a ground-dwelling one, Epigaeus, and one that looks like little fir trees called Abietinus. We also have a very rare one called Adenophorus periens. Now this one was last seen, it was known from the Big Island to Kauai, and then it just disappeared from the colonies that we knew of. Didn't know of any in 2014, and it was back in 2014 when one of our biologists, Shauna Walsh, and one of the PEP folks, Scotty Heinzman, the PEP, the Plant Extinction Prevention Program of the state, discovered some young plants in Wainiha Valley, and subsequently the state discovered some more in several other colonies on Kauai. So we do have another chance to protect this unusual fern. Here you see it pendant, and it's unusual Venetia blind-like pinnations. So yeah, so it was thought to be extinct, rediscovered. Now we're trying to learn, and Ruth is spending a lot of time trying to learn how to cultivate these green spored ferns. In the Doryopteridaceae, we have quite a few interesting plant species and genera: Arachnoides, Tinnitus, Sertomium, the holly ferns, the Elaphoglossum with the sporangias completely covering the fertile blades. They'll have dimorphic blades, some just sterile blades, and some that will become fertile. A couple of interesting ones is Doryopterus cornalis podosaurus, described by Tim Flynn, our herbarium curator. Here you can see the stalk, why it was called Podosaurus. It's got its sporangia on a foot. Here, there's maybe 30 or 40 individuals of this plant known, and we've been gathering spores of this unusual fern, this Doryopterus, and trying to cultivate it. Another interesting one is the Tinnitus. Tinnitus squamidra was known from several islands: Molokai, Maui, Oahu. It was historically known from Kauai back in 1898. It was collected by Amos Heller, and then not seen again until just a few years back when, oddly enough, a plant was found in the northwest of Kauai in some mesic forest. And then pretty much that same month, in the next valley over, another second plant was found. And so there it is, Tinnitus squamidra, and there it is, the importance of doing field work and knowing a little bit about the history of where these plants were found historically, and where to look for them, taking the time to collect fertile spores, bringing them back, trying to cultivate them. It all takes a lot of time and money. There's not enough people to actually carry out all these chores. And so we're hoping that more people will become interested in this type of research. Philipteridaceae is another interesting family in Hawaii. We have quite a few, I think 8 taxon here, 8 taxa here. One of the rare ones is Menisciopsis wailele, which once again was described by Tim Flynn, our herbarium curator. Very interesting one. It's a rheophyte, one that likes to be on the banks of streams and can handle extremely fast-moving water. It's known from around 400 individuals, and they occur in very remote areas. Menisciopsis cyathioides is one of the larger examples of our Philipteridaceae, a long, erect rhizome with very long stipes. The underside of the blade demonstrating the sporangia. Another very rare one is named Menisciopsis boydii. Some people might notice Cristella boydii or Philipterus boydii. The names have been changing. And this is just showing the importance of phytogeographical information. These are the locations on Kauai where this Cristella or Menisciopsis wailele occurs. And like I said, they're in remote streams. And so we do utilize satellite GIS information to clearly map where they are and give potential for those interested in conserving them with their permits to go and try to collect and to cultivate them. Here you have an example of more of a creeping rhizome. And recently a new species was discovered in the remote areas of Maui. Very steep, very tight drainages. Once again, a rheophyte occurring on these side walls was Ethereum haleakali. The Hawaiian Islands only knew of one species of Ethereum microphyllum. But here, this thing here, as you can see, this is 1 centimeter long. It's a very tiny one, whereas the microphyllum, the previously known one, can be over a meter or so in size. So it was hard to see, but nevertheless, with someone who would be familiar with all the different species would stop and look at it and say, hey, wait a second, this is an Ethereum, and we don't know any tiny Ethereum like this. And so hence, Ethereum haleakali was discovered in the remote areas of Maui. So I'm going to be closing this talk now, with 180, approximately 180 species of Hawaiian Lycophytes and ferns. I can't really spend too much time covering it all, but in general, there is, like I said, a great need for conservation efforts to be made for protecting these plants. Some of the current goals that we'll be focusing on include conducting regional surveys, and once we do discover or document rare plants, we'll either describe the new species or publish the phytogeographical data so people can make efforts to preserve them. And then, like we did with the Esplenium diolysiniatum and the Esplenium diolmanii, reestablishing populations of these rare plant taxa into in situ or in natural areas where they originally occurred. And I think that's the end of my talk. Yeah, so I'd like to close the talk. I'm going to escape this, stop share.
Thank you, Ken. Appreciate it. So we had a couple of questions come in that I would like to ask. If you want to go ahead and share your video so we can see you, that'd be great.
Alrighty.
And if anybody else has any questions, feel free to drop them in, and we'll do this again at the very end as well. So we had one question. Janet was wondering about tips and tricks or resources for IDing ferns. Nina suggested to her Daniel Palmer's book, Hawaii's Ferns and Fern Allies. But do you have any other recommendations for her?
Yeah, so that is a great choice. Daniel Palmer's 2003 publication of the Ferns of Hawaii. Amanda Vernon, who was worked actually here for a while, and so she took an effort and put together a key that you can go online and you can go ahead and ID some of these ferns online, either vegetatively, do it that way. So yeah, I would recommend that. And there's yes, so those are some of the most current literature that would help.
Thank you. We had another question. This one is from Amanda, and she wonders, is climate change having a measurable effect on the ranges of these ferns?
Oh, absolutely. Yeah, climate change, things are heating up. Some of the Hymenophyllum that we talked about, some of those single-celled ferns, The filmy ferns will and are being affected. When you go into some of these wet forests already, I'm seeing a decline in that family group. And so those being some of the more sensitive, I think that might be just the beginning and as an indicator for what might be in store. So yes, climate change is already showing its ugly head here in Hawaii.
So I want to ask one more question, and then we'll pass it over to Mike DeMotta, and then I'll come back to questions at the end. So David is wondering, do you have any, and I apologize for my pronunciation, Equisetaceae in Hawaii?
Yeah, no, we don't have any in that group, I'm afraid to say. I guess it was hard for them to make their way over here. So no, we don't have any native species in that group.
Well, thank you, Ken. I appreciate it. So like I mentioned, we'll come back to questions at the end, but I want to pass it over to Mike DeMotta. And
before I go,
go
can I just say one more thing?
Sure.
As far as references go, I'd also like to mention Tom Renker, who has also, he's probably one of the great 21st century teratologists, and he's written an abundant amount of information about Hawaiian ferns, and I tip my hat to him. And so if you do a little bit of a literature search for Tom Renker, you'll come up with a lot of information on Hawaiian ferns. So I'm sorry to interrupt you.
Oh, no worries. Great. Thank you, Ken. So we will pass it over to Mike DeMotta. He's our curator of living collections, and he's actually going to give you a live tour of the Fern Lab. So it might take a second. Let's go ahead, Mike, if you want to unmute yourself, we can test it out, make sure we can see and hear you.
Sure. Can you all hear me?
We're good to go.
Hear me good? Okay, great. And just so that everybody knows, I'm fumbling with a new device here, so if it starts spinning around, please bear with me. Again, my name is Mike DeMotta, and curator for living collections. I am at the Conservation and Horticultural Center in Laua'i Valley, the home to our nursery. And I'm down here because we're going to do a live tour of our Fern Lab. And so for anyone who's volunteered here before or had the possibility of coming on a tour through McBride Garden, you would have seen our facility, and what we now use as our Fern Lab initially was designed to be a micropropagation facility. Early on in my work here, I was participating quite a bit with some of the restoration work that was going on in Limahuli Valley and Preserve. And Dr. Ruth had already started her research on some very rare fern species, the ones Ken was talking about, the Dialia. And one of the things that occurred to me and we talked about was how important ferns are in the habitat. Initially we were looking at what kinds of trees and shrubs we might need to do restoration work in Lower Limahuli Preserve. But having followed Ken through some beautiful habitat all over Kauai, it also occurred to me that part of the habitat restoration must include ferns. And at the time, our skills only allowed us to grow ferns for the most part from rhizome cuttings, as divisions and that sort of thing. And there's really a lack of diversity in the genetics when you reproduce any plant clonally that way. So we had talked about it, Ruth and I and others, and Ruth had been researching growing Dialias from spores. She was in the middle of her research for that. So she helped us get our first areas of fern propagation going. We were using our what we call Acacia Lab, which was located up in our main administrative building. And it was all really new, but there was a lot of early success. This was back in 2006, 2007. And we grew quite a few ferns over time and were able to send many of them out to Limahuli Garden. So years have gone by and we've been continuing to grow ferns in our now dedicated Fern Lab. And still though, we generally were using part-time assistants through our nursery managers, through others who were working in the nursery, volunteers and interns. And the difficult part of that is an intern project or intern program may have lasted for a year. So every year we were basically having to retrain people on all the procedures and protocols that take place in our fern lab. And so in the last few years, the fern propagation has taken the front stage in importance. We all realize here at NTBG that, again, ecosystem restoration means everything from the top of the forest right down to the ground. And, you know, if you've been around some of the high mountain peaks in the Hawaiian Islands, like here in the Elaka'i Plateau here in Kokee, you know, the pristine areas are covered in ferns, lycophytes, you know, the terrestrial ferns, epiphytic ones. It is what keeps all of that rain that we're famous for in the middle of the island at Waialeale from simply running off the surface of the island. The ferns and everything else that is growing on the ground there help to keep all of that in the ground, absorbing it all like a giant sponge and then releasing it slowly. So the importance of ferns in the ecosystem cannot be overstated. The flip side of saying that is if ferns and other epiphytes were to disappear from our forests, especially in the watersheds, I think in the long term, we wouldn't be able to live on the island anymore because we would not have that ability, the islanders would not have the ability to retain the water, the moisture, and release it slowly into our streams and into our wetland areas and so forth. So ferns are as important in the ecosystem as everything else. It's all integrated and tied in together. So since we at NTBG and in the nursery have focused on rare plant conservation and propagation over the years, we have taken on and made it an important goal for us to do what we can to conserve rare fern species by learning how to grow them, each species one at a time, from spores. Spores collected in the wild will bring us the genetic diversity that's really important in the long-term conservation of anything. And interesting, one of the comments I've had in the past were, well, you know, most of these ferns grow on their own in the wild without anybody's help. So why do we need a fern lab to do this? Well, some ferns have become very, very rare, and they need our help to reproduce because in the millions of years that the Hawaiian Islands have existed, many of the animals, for example, like feral pigs, goats, and deer, did not exist. So the ferns did not have that kind of competition. There were no browsing animals, so to speak. So they do need our help to recover their numbers. And so we've set up the Fern Lab, and I'm going to take you on a little bit of a tour of the Fern Lab. One of the things about the lab is it is air conditioned, maintained about 64 degrees. Many of the ferns we're growing, especially the really rare ones, come from higher elevations. So the cooler temperatures sort of replicate the conditions in the field, in the wild. And then it helps to reduce the possibility of pathogens. You know, nothing worse than moist, warm air to get fungi and other diseases to grow. So let's flip this around for a second. Oops. Sorry, everything is turning around for me. Here we go. All right, so we're now going to walk around the Fern Lab. It's a two-room facility. We have microscopes, and we have our hood, and then we have all these racks with all of these phyta trays, which are growing trays where we put our potting mix and plant our spores. And I'm just going to walk us through to the other room. You'll see. I'll remove it.
Yeah, maybe if you're having trouble, you can just take it off of the gimbal and hold it.
Yes, I'll try and remain steady. So as we walk through, we'll see all the growing racks with the grow lights. Grow lights stay on about 14 hours a day. That is to provide the ferns, the young plants, with the ability to photosynthesize. And also the length of time that we keep them running also helps to suppress the growth of algae. So you'll see we have two kinds of growing lights. The broad spectrum one, and then we have these LED lights that have blue and red spectrum. Let me take you to some of the trays that we have in the lab. Well, so first of all, when spores are collected, as Ken was talking about, Ken's talk, by the way, covering so many species of ferns found on all the islands, was really, thank you, Ken. That was a really wonderful talk. And Ken is one of our field biologists that will we'll find plants, ferns that have mature spores, and we'll collect them. It's really important to prevent cross-contamination. So spores generally are pressed between sheets of paper, the leaf blades are, so that the sori will dry and the spores will be released. We then use a sterile media. The media we use, the planting mix, is a mixture generally of, we will sometimes mix with native soil, and then we will use a commercial mix that can be a blend of peat moss and cocoa coir, mixed in with a little vermiculite to retain moisture. So this is a newly planted culture of fern spores. So the fern technicians that we have will come in here and look at each little container like this and determine whether or not it's ready for being watered. We do not water everything every day all the time. We judge how much water each container needs by physically observing. So the next step is the fern spores are germinating. So here's a nice tub where the spores have just started to sprout. And if you were hiking in the field, you look down and you would immediately assume that was moss. And especially when it gets into this phase, right? So these are the gametophytes. That's the next step. They're fully developing. They're beginning to exchange. And the next step after the gametophyte phase will be sporophytes. And you can see that's starting to form in this tub. This is the next step. And finally, the sporophytes form where you can actually discern the individual plantlets. Ken showed a wonderful picture of Esplenium nidus, which is commonly referred to as the bird nest fern. It is an indigenous species. It is found on various parts of Kauai. And these are one of the plants that we've been growing in large numbers to be transplanted out at Limahuli Preserve. And here's an individual that is just about ready to leave the fern lab and go out into our greenhouse. In the greenhouse, we have a section that we call the cool room. There's a swamp cooler fan in the cool room that turns all day, and it keeps that particular room a little bit cooler than the rest of the greenhouse. And our ferns are then hardened off in the cool room. So to walk you through, we have ferns of different sizes in the lab. So Doryopterus fusco-atra, we have Doryopterus decora, a lot of different species, and some of them are from the wet forest, some of them are from dry forest. We have a couple of different species of dry forest ferns that, as Ken pointed out, a few pictures of Doryopterus angelica and Doryopterus decora that will go dormant in the hot summer months. And so we will be able to harvest those after a wet rainy season when they become active and they mature. We're able to get spores out of those, and those have been really easy for us to grow in the sense that they are well adapted and we have the protocols for them. Now this bench here, I'm going to point out the sign here. It says Fern Spore Project. A new project that we recently started is in our herbarium where we have all of our dried specimens. We have two of our interns going through and looking at all of the fern collections and making determinations as to whether or not there are viable or usable spores within those fern collections in the herbarium. They're recovering spores from that and sowing them here in the fern lab to see if spores retain their viability after going through the herbarium process, which includes drying out of the plant material. To use as a comparison, when possible, fresh spores are collected of the same species. So we can use the fresh collection sort of as a control and see how it compares to with the dried specimens that are coming out of the herbarium. The goal is to determine that if some ferns have viable spores after going through the herbarium process, with luck, we may be able to recover fern species that have become rare over time. And this is important to know because as the question raised a little bit earlier about climate change, some of the species that have become quite rare, if we're able to recover viable spores from our herbarium, that would introduce a genetic profile that might be lost because those populations have declined or gone extinct altogether. So the Fern Lab itself is not very big, but the way we have it set up, we take as good advantage of the space that we've got with all the different shelving units. Yeah, I'm not sure if anybody might have any questions.
We sure do have some questions. So we have one from Amanda. So she works on Midway Atoll, specializing in native plant propagation for habitat restoration. And they have historical records of Psilobium nudum, N-U-M, being present out there, but it has since been extirpated. Are there other native fern species that can tolerate conditions, salt spray, dry summers, coralline soil, on coral atolls that might be useful for coastal restoration?
Well, that's a great question. On Lehua Island, which is not a coral atoll, but it is still a very dry coastal island just west of Kauai, we've done a lot of work on Lehua Island since before 2007. Ken did a lot of field work surveying Lehua, and one of the plants that they found there is the native Doryopterus decipiens, and it's one of the Doryopterus that will go dormant during the summertime. Lehua was famous for having rabbits introduced there, but the ferns were able to dodge the rabbits by going dormant in the summer when all the other food supplies were disappearing, so it's still there. So I'm not sure about the sandy conditions at Midway, but I do know that Doryopterus decipiens, which is found in coastal sites on most of the main Hawaiian islands. I have seen it in the Waianae Mountains on Oahu, for example, and here on the south shore of Kauai, is something worth considering.
Thank you, Mike. I'm going to jump over to Ken for a question from JD. So they, first of all, mahalo for the great presentation. I'm curious about how invasive ferns affect native ferns in the environment.
Oh yeah, so invasive ferns, like some of the Blecnaceae family, we have Blechnum appendiculatum, we have Adiantum hispidulum. They will completely cover, and they're very vigorous, they'll completely cover terrestrial areas and displace our native ones. We don't have that many invasive ferns in wet forest areas. There's a few. We worry about the mule's foot fern, for example, that can get big, but there's never great numbers of them. We have certain in the Polypodiaceae, we have certain members that'll spread here and there. But the real bad one is in these mesic and dry regions where these Blechnums and Adiantums will completely cover the ground and are a real serious impediment towards establishing native cover.
Thank you, Ken. And here's a quick one for you as well. Somebody was wondering, are staghorn ferns native to the Hawaiian Islands?
No, we have no native staghorn ferns here in the islands, but they are beautiful. And yeah, nope, that's a short answer.
Thank you. So I'll jump back over to Mike DeMotta. So let's see, we have Janet here was wondering, how long does it take from planting quote unquote spores until a fern is ready to plant out?
Oh, that's a great question. And, you know, it varies from one species to the next. And I'll just go back to some of Dr. Ruth's early research with Dialia pallida, which when she started working on that, I remember hiking with Ken Wood into the Northwest Valleys here on Kauai and only seeing a couple of plants in the wild. And so Ruth spent several years collecting spores and trying to figure that part out. And one of the interesting things was she learned that the spores from individual plants had to be mixed together for the gametophytes to be able to reproduce. Otherwise, they'd stay in the gametophyte phase and never grow. So that took up quite a bit of time. But even when she figured that part out, it was still nearly 4 years for that really rare fern to go from spore to sporophytes and then to plantlets that were big enough to go out. So it was quite a long time. But again, you know, when you think about the isolation of the Hawaiian Islands, all the plants were kind of in the niches that they evolved into. So they didn't need to compete very much against other plants that were here at the time. Other ferns, for example, and the Dialias grew in their ranges and did not have to outcompete each other. As Ken mentioned, some of these introduced species are super competitive. And then trampling and habitat destruction was really had a big impact on Dialia. Some of the others, though, like are common and very important lay species, Polypodiaceae, we call that in Hawaiian, which is very important in Hawaiian culture and used in lays. That can be ready to go out in a matter of months, in a year or so. We've grown the endemic Satellaria sathioides, Mau ferns, and they've been big enough to be sent up to Limahuli Garden in about a year's time. So it varies widely from one species to the next. And that's part of the point of running the Fern Lab the way we are right now, is to learn all of these protocols and all of the results so that we can know if we get a new species or a rare species in a genus that we have experience with, it'll give us some background on how best to grow that.
Thank you, Mike. I have another one for you from David. Do you cultivate lycophytes as well?
Not yet. Ideally, we'll be able to do all of that. I have to say, as I think I mentioned a little while ago, that manning our Fern Lab has been primarily done with spare time from our nursery manager, volunteers, and short-term interns. We recently got some funding from a grant proposal that we submitted, and NTBG has committed additional funding where we are able to hire for the very first time a full-time Fern Lab technician. That will really open up the doors for us because we'll have somebody fully dedicated to everything that takes place here in the Fern Lab. And so we're really excited for the potential for what we can learn and what we can grow, and the amount of fern conservation that NTBG will be able to do in the next couple of years. So our funding for the lab is for the next 3 years, and we're going to be working on finding additional funding to continue this for as long as we can.
Thank you, Mike. And I know some of you who are on here are some of our fellows. So that is our fellows project this June, is fundraising for the Fern Lab in this project. And I'll drop the link into the chat if you guys want to learn more about our Council of Fellows. Sorry. So we just have about a minute left before I close out. Ken or Mike, do you have any closing words you'd like to share before I do some housekeeping at the end here?
I don't mind making a statement. Well, it would be basically that, we're on the third planet from the sun. It's an incredible planet. We've been given a gift. The diversity is one of the miracles. It's one of the wonders of the world. And each species is a masterpiece of creation. Let's get it together. And I would like to see and more funding coming through. I see a lot of interest in politics and sports and other things. It would be nice if we had a renaissance in conservation, and whereby people take it real seriously. We at the NTBG could really use some support to manage even just one species, and to be able to collect fertile parts, spores or seeds or whatever, to keep a plant going. Even just one plant you want to get from several individuals, and to manage each one is difficult. So I'll cut it short here and just say, please help us if there's if you can spread the word. If you know of some organization that wants to get involved, get in contact with NTBG, and please help us to preserve our diversity here. Thank you.
Thank you, Ken. I appreciate that. And I'll also share a link to a recent news story we put on our website. That's a really great success story about our scientists going out in the field and finding seeds from plants that are critically endangered and having success in propagating at our nurseries and outplanting. So thank you, Ken. Appreciate that. Well, Mike, if you are good to go, I'll go ahead and close it out. Just wanted to thank everybody for joining us again. And I will be sending out that email with this recording. And as Ken mentioned, please feel free to share the recording with your friends and your family who you think might be interested in learning more about ferns and about our conservation and restoration work. And you're more than welcome to reply to my email if you guys have any questions that didn't get answered or a question that you thought up after this. I'm happy to follow up with you there as well. And thanks again, everybody. Hopefully we'll see you on June 1st for plants along streams and coastlines. Mahalo.
Thank you, everyone. Bye.
Thanks.